Today, we report on studies presented at the 2016 ASCO Annual Meeting that highlighted developments in precision medicine. Welcome to Enclave News Network. I'm Gina Columbus. A research study launched in October 2015 could help expedite metastatic breast cancer genomics research, as well as provide leads for developments of new treatments. In the 7 months since the project began, more than 2,000 patients have enrolled in the project, designed to collect and analyze health records, tumor specimens, and saliva samples from patients with advanced disease. With the project, which was launched by the Broad Institute of MIT and Dana-Farber Cancer Institute, the website mbcproject.org was developed to recruit patients directly into an ongoing trial. 95% of patients to date have provided information about their cancer, treatments, and their experiences. Commenting on the results, lead study author Dr. Nikhil Weigel, a medical oncologist at Dana-Farber Cancer Institute, said, "In the United States, only a small fraction of the 150,000 women and men living with metastatic breast cancer receives care at centers that conduct research on tumor samples. With this new approach, we hope to engage patients to directly participate in research, regardless of where they live, and contribute to the effort to improve outcomes for all people with advanced breast cancer." Patterns of genetic changes detected in liquid biopsies closely mirror those identified in traditional biopsy, according to a study presented at ASCO. This is one of the largest cancer genomic studies ever conducted, as samples were obtained from more than 15,000 patients and 50 different tumor types. The study assessed the accuracy of liquid biopsies compared with tumor samples through patterns of genomic changes in circulating tumor DNA, as well as the consistency in the frequencies of particular changes in ctDNA against previously published data from genomic analyses of tumor tissue. Commenting on the results, lead study author Philip Mack, professor and director of molecular pharmacology at the University of California Davis Comprehensive Cancer Center, said, "These findings suggest that analysis of shed tumor DNA in patient blood, also known as a liquid biopsy, can be a highly informative, minimally invasive alternative when a tissue biopsy is insufficient for genotyping or cannot be obtained safely. Moreover, this test, known as Guardant 360, provides an unparalleled opportunity to monitor changes in the cancer as it evolves over time, which can be critical when patients and physicians are discussing treatment options for continued tumor control." A Phase 2 study that matches patients with molecular abnormalities in the tumor to corresponding targeted therapies has shown promising early results. The nationwide MyPathway study is an ongoing, non-randomized open-label trial that evaluates 4 treatment options for patients with advanced cancer for whom no beneficial treatment is available. Patients who must show abnormalities in the HER2, BRAF, Hedgehog, or EGFR pathways are matched with drugs targeting these abnormalities. HER2 patients receive a combination of trastuzumab and pertuzumab, while BRAF patients receive vemurafenib. Hedgehog pathway mutant patients receive vismodegib, and erlotinib was given to patients with EGFR mutations. A total of 29 patients with 12 types of cancer responded to targeted treatment, and the most promising efficacy was seen in patients with HER2 abnormalities. Commenting on the study, lead study author Dr. John D. Hainsworth, a senior investigator at Sarah Cannon Research Institute, said, "With genomic testing of tumors becoming increasingly available, studies such as ours will help more patients benefit from precision medicine approaches. Although it is still early to draw conclusions, our findings suggest that, for example, HER2-targeted therapy could be expanded beyond the current indications of HER2-positive breast and gastric cancers. Our study gives strong early signals for activity of HER2-targeted therapy in HER2-amplified colorectal cancers, those with extra copies of HER2 gene, and possibly other HER2-positive cancers." That's all for today. Thank you for watching Enclave News Network. I'm Gina Columbus.